In today's session, we'll pick up from where we left off last time. We had a little bit of difficulty completing some of the things in Chimera, and then later on I had a chance to talk to some of you that were using different software or had trouble with Chimera, and so obviously that's something that we need to think about, how we can help everyone understand the relationship between the sequences and the protein structure, and think about different properties of amino acids, which is an important part of being able to interpret the analysis that we've been doing up until now that pointed to different types of origins, potentially the way of transmission, and could help characterize some of the disease pathology that we observe these pathogens are causing. Today, we'll talk about a different aspect of mutation variants, which is rate of mutation, and how we can characterize mutations from the standpoint of their fitness, how mutations actually help the virus cause disease and find a balance between efficient immune response causing the suppression of the immune response, and then also helping it to replicate. Of course, a lot of this is, we will see probably today. I hope everyone can appreciate how, in reality, little we know about the way viruses work. Some of the viruses are model viruses and have been studied extensively for a long period of time. Many of the viruses have been the focus of data generation for decades, and some of the studies, of course, have limited impact because we still have so many unanswered questions about these pathogens. We'll see, I think we already saw how, a lot of this is trying to piece together a puzzle with a lot of different sources and a lot of different types of information. And that is a huge challenge that I think requires a certain perspective from anyone engaging in these kinds of bioinformatics analyses, that essentially we are on a quest to find information, characterize that information in terms of its reliability and how much information it can give us, and kind of combine the data with the biological knowledge that we have from experiments, from an understanding of how the virus works, relying on the data generated by specific types of experiments. So before we continue, I am excited to see that there has been a lot of progress. There's been some interaction on the forums. I saw that just as recent as today, right before the session, we had a few different questions posed on the forum. Because it was right before the session, I did not have time to respond. But I do want to point out that I saw several of the pipelines that some of you have been running. And you have to keep in mind that even if it's a somewhat simple task like multiple sequence alignment, if you have hundreds of sequences and multiple groups, Lashanda, I'm talking about your pipeline, it's going to take time. So don't expect, to analyze huge amounts of data and get your results overnight. The more data you have, the more complicated the alignment becomes, because multiple sequence alignment seeks to align those FASTA files, those sequences, in such a way that they are optimal across the full sequence, so minimal number of insertions, as well as maximum likelihood of those alignments happening in the correct places. And there's multiple factors to consider there before that alignment is performed. And yes, we are going to put up, we'll talk more today about the independent project, and hopefully some of you will be able to start thinking about the project, submitting some of your ideas, because really what we need to have is a discussion about these different project ideas. And today I'll kind of offer you some of these project ideas as well, so that we can think about how these projects could be done together. Ultimately, as you will see, a lot of the effort goes into being able to identify a question, find the data, but also an equal amount of time will be spent on the actual analysis, maybe many different times until you get something meaningful, and then the interpretation. So having to do all of this on your own is time consuming and requires a lot of focus, and having multiple people work on the same problem is going to be much easier and faster. Okay, so let's review some of what we did last time. Last time we talked about the human infection, the origins, and so I wanted to kind of put together visually how these different elements actually correlate with each other in terms of the different types of mutations that we will continue to talk about today. So essentially, at the start of transmission, we're talking about a single source or the origin of a specific strain of a virus, a variant of that virus that we think is the origin of what we observe in human population. One of the, so that specific event is called the spillover event. So zoonotic transmission happened potentially just once, right? So it came maybe through an intermediate host, but ultimately it came that one time. So a big concern moving forward from that single spillover event is actually having an intermediate host, and that intermediate host might be in close proximity with the original host, where there might be a different dynamic in terms of evolution, because maybe the immune response is very severe, maybe because there's a lot of other competing viruses. And so in that evolutionary competition situation, a lot of novel mutations, a lot of recombination can happen. So having an intermediate host that begins transmitting viruses repeatedly is a big concern. And one of the bigger concerns in all of this is having a good affinity between the viral proteins and the host receptors, the host proteins. So then we also think about the process of viral adaptation to a new host. And so the initial adaptation is that ability to enter the cell. The receptor binding is just the key getting in. But then all kinds of additional variations start occurring because of the immune response. And so you can see here the protein interferon gamma, which is one of the cytokines that is involved in the response, in the cellular response to this virus entering. And so then you have the dynamic of the virus evading now specific to this host or specific to this tissue even, immune pressure that occurs and might be very different from what happened in the previous host where it originated from. And eventually, there is a process of adaptation that also causes perhaps, or one of the evolutionary objectives of the virus is to be able to adapt to different tissues, adapt to the different hosts and the transmission patterns. And all of this is at the same time under the impact of the stochastic process, random process of mutations occurring over time. Because it's a single strand RNA virus, because RNA has more mutations just naturally, it's a less stable molecule, and we have a lot of random mutations that occur. So now when we think about how do we fit in our analysis of multiple sequence comparison and mapping of those differences on protein structures, we need to consider all of these different processes. And so considerations about the nature of these processes, some of them being stochastic. If you think about, for example, tissue transmission, how the virus goes between the intranasal cavity to esophagus to the lung and alveoli, so that process is also not going to be exactly the same between different patients and between different hosts. And there are many kind of specific types of processes and factors that are, to be considered there. And so when we think about these different similarities between sequences and where those similarities or differences are found, we have to map them to our understanding of how the virus is governed by these different types of responses and processes, because that's essentially the only way that it can respond is by modifying the sequence. And so when we have an overall kind of a high level view of similarity between multiple sequences, which we can map by aligning these sequences together and mapping them onto an evolutionary tree, which we've done before, one of the questions that we can ask is, high level organization, how these might be related to each other, and potentially what are some common or different origins of these different viruses. So one of the important things when you consider this question is to think about whether you can trace multiple spillover events, understanding that you don't have all of the sequences that you might need, right? So when you consider, for example, an intermediate host, having the data from dogs, from cats, from cows, from the different animals in close proximity to the human population, as well as having a variety of different origin sequences, for example, multiple different bats, all of these things are going to be important as you think about the process of origin and evolution in the human population. So this kind of analysis is highly dependent on the types of sequences and the representation of the different factors that you want to consider in that process of development. Now, the other approach is once that picture is somewhat clear, how do we use this data to be able to answer specific questions about each one of these stages or events? For example, one of the things that we did over the course of the past couple of lectures is think about how can we rule out some of the proposed theories, hypotheses about the origins, for example, the virus being intentionally manufactured or modified to cause higher virulence and severe pathogenesis, and how can we understand the differences between known sequences of viral genomes from the SARS-1, MERS, and SARS-2 in terms of potential sources, right? So one of the things to remember here is that a lot of these things are not conclusive because of lack of data, because of lack of experimental validation, and because some of these things are really affected by these multiple processes that we described in that one slide. So when we think about this, here we focused on specific areas of that key protein, the spike glycoprotein, where we looked at mutations in the receptor binding domain and the polybasic furin cleavage site and O-link glycans, where we talked about how these actually help us understand the binding of the spike glycoprotein to the ACE2 receptor, the cleavage of the S1 subunit, and then cell fusion, which is where the S2 subdomain is playing its major role. And then we can also think about the glycosylation sites and how those are different between different proteins. Those might be responsible for both the way that it binds to the receptor, as well as potentially immune evasion, because glycan, these molecules, sugar molecules, glycans, are actually protecting some of the areas on the protein from being recognized. So the first part that we did was we looked at this polybasic cleavage site, and the conclusion was that we see an insertion of a specific sequence. That insertion is fairly consistent in all of the novel coronavirus SARS-CoV-2, and it is different from the bat coronavirus, it's different from the pangolin coronavirus, and it's different from the SARS-1 coronavirus. And so the conclusion from that polybasic cleavage site insertion is, again, now a hypothesis on whether that is helping the furin cleavage. And it has been experimentally shown that if the cleavage is more efficient, then there's a higher ability of this virus to enter the human cell and for cell fusion. So it can not only enter the cell one time, but also go between cells without getting outside of the cell by fusing membranes of two cells together. So these kinds of conclusions, again, are highly dependent on our understanding of the structure of this spike glycoprotein. And so we looked at how the specific cleavage site being exposed might explain how we can imagine this furin being able to more easily cleave the S2 portion of the protein. Now, the other part that we talked about was the receptor binding domain. And what we wanted to do was really use our understanding of these different portions of the virus and seeing the sequence of that specific receptor binding domain, as well as the receptor binding motif, which is the actual certain number of about 15 different amino acids that actually come very close to the ACE2 receptor, to understand how that might change the affinity or the how well it binds to the ACE2 receptor. Okay, and so a question, is the insertion natural or hypothetically a genetic manipulation? So it is true that there have been specific modifications to sequences that are known to cause a specific effect. It's true that you know, because of research, you can now try to introduce those, and it can get out of hand. So one of the explanations in that paper that we're discussing, and hopefully you had a time to take a look at that paper, is that the idea of a specifically engineered virus, right? So a virus that was engineered to produce a certain effect, obviously is a reasonable assumption, right? So in research, that's what virologists do all the time. They try different things. They can, you know, try to hypothesize whether this will cause this kind of phenotype or that kind of phenotype, and then test it. So from that perspective, it's impossible to rule out how that specific polybasic cleavage site was introduced. Because we don't see it in any of the original hosts, right? So it's not seen in bats, it's not seen in pangolins. We assume that that kind of an insertion would happen either in a virus that, right? So let's go back over here, right? So you can see that even the closest sequence from the bat does not contain this insertion. And then in the human viral population, we see that insertion very consistent. So the question is, when did this insertion happen? If it did not happen in these other hosts, then when did it happen, right? And so I think that's a reasonable question. How did it appear there? The other thing that is seen here is the differences in O-link glycan residues, and there's also a change, right? But that change is much more consistent. So we know that glycan residue changes change affinity, change effectiveness of binding, change immune evasion, and that seems to be a much more natural progression from these hosts, established hosts and potential intermediate hosts. So I don't necessarily have an answer on how that polybasic cleavage site was introduced. Could it have been introduced naturally? I think to have a conclusive answer to that, you will have to find sequences that contain the exact same polybasic cleavage site and the exact same portion of the protein, and then you will know for sure that happened naturally, and here's an evidence for that. Could it have been introduced, you know, artificially? Obviously, it could have. Was it done to engineer a specific virus that causes these kinds of pandemic effects? that is questionable because there are specific platforms to produce viruses, and this specific virus is not based on any one of those platforms. So in other words, this was not a specific engineering exercise to engineer a virus that somehow could be mass produced and used for some kind of a malicious purpose, specifically because of this cleavage site. So I think, there are still unanswered questions about how it got there. And, obviously, if it got there, how did it then spread between humans? But, we don't have any evidence of malicious intent to engineer a virus with this specific polybasic cleavage site to cause some kind of a pandemic. So I think those are, some preliminary answers. And I think if you are really interested in that kind of a question of whether this could have been a naturally occurring insertion, one of the answers that you would want to kind of support your answer with, one of the exercises that you could do is find additional coronavirus viral strains from different hosts, from different bats, from different other animals, right? We're not just talking about bats, we're talking about coronaviruses being, in pets, being in meat, being in all kinds of other animals around us. And so we would have to really look for those sequences in all of those viral strains to maybe find evidence of that being present in other animals. Okay, so if we now look at this receptor binding domain, right, one of the other questions that we have is whether the specific sequences that actually bind to the host receptor could have some different physical chemical properties, right? So for example, one of the things that we do know is that if we take all of the different sequences from different coronaviruses, in general, that whole region is more variable, right? And so previously we looked at that variability between different hosts, so bats, humans, pangolins, etc. And also we can look at even humans, right? And we see a lot of changes happening specifically in the S1 subunit in general, but even more specifically in this receptor binding domain. So if we now do a comparison, right? Again, if we just take the spike glycoprotein, let's forget about the rest of the genome, and we look just at the spike glycoprotein, one of the things that you can do is select certain spike glycoproteins and align them, again, producing a phylogenetic tree. And this is a phylogenetic tree that I did to prepare for the session, where you can clearly see that there are some bad coronaviruses that are not related, and then this bad coronavirus RAD-G13 is much more closely related to the human SARS-CoV-2. You can also see that pangolin coronavirus is actually in the same clade, so to say, right? Even though it's not as closely related, but all of them are very different from the original SARS-1 and the Hong Kong SARS-1 specific strain HKU, right? So these are different from other coronaviruses. If we look within the human population, so just after the COVID-19, we can identify very specific changes. So in other words, these are not random mutations that define this similarity. These are kind of specific changes. And you can see on the amino acid level, right, this is position 614, a change from a D in the reference genome to a G, a glycine, in these specific human strains. And also, if you do a comparison between the RAD-G13 and the COVID-19, you can also see that the mutations do not arise in some random spots. They arise in some very specific spots. And these specific spots, I think, is what we want to explore today, what we wanted to explore actually last time, but we're going to quickly go over this today so that all of you have a sense of how to apply specific tools and methods to start looking very closely at some of these residues that are responsible for the contact. Okay, so how can we do that? First of all, just to kind of give us a general biological background, this is the ACE2 receptor, and there's a specific structure that we can find on the protein data bank, 6M17. And this 6M17 is a structure that has both the ACE2 receptor, which is just this blue portion. It has the B0AT1 receptor, which is these two gray portions. And then it also has the receptor binding domain, the part of the spike glycoprotein that was actually taken together in this model. And here you can see some of these, some of the kind of logic behind how this spike glycoprotein works with this receptor binding domain coming up from the overall spike glycoprotein, right? And so you can see how the frequency of receptor binding domain seems to be lower in SARS-CoV-2, right? And this might be the result of the pre-activation by furin, or might be a result of how the structure itself, the chemical properties. So if you see that it is down, it seems to evade the immune recognition and response, right? Because essentially this top part is the one that is exposed and easily detectable. And also it contains some specific residues that are important for the binding, right? So if that section is recognized by the immune system, then the whole virus is exposed. And then you can see that the human ACE2 binding affinity, so now the specific residues in this portion, those seem to have a higher affinity in SARS-CoV-2. Some papers propose that it's significantly higher. Some papers say that they're not that much higher, but still a little bit higher. But nevertheless, this might be an interesting component that we can look into. And then pre-activation by furin is not a part of the SARS-CoV, the original SARS-1 mechanism, but it is a part of the SARS-CoV-2 mechanism, and which might make it more effective at connecting. So let's take a look at how this actually works. The membrane of the cell has a specific opening, and that opening is where this ACE2 receptor sits. And the ACE2 receptor then has, so this is the BO81 sodium-dependent neutral amino acid transporter. And then we have this receptor binding domain. So here we just see this individual piece, but essentially this piece belongs to this receptor binding domain portion of the spike glycoprotein. And that's a part of the virion. So essentially what we have is this. One of the first things that we can do, right, to study this is to try and connect the spike glycoprotein model together with this ACE2 receptor model and identify what we see right here is the receptor binding domain, which is highlighted right here between these positions, so this region. Okay, and so the exercise that we were trying to do, and I'll show you kind of quickly how to do this, is the ability to use the 3D structures and the sequence alignment to map these structures to each other. Two examples that I want to quickly show you. Hopefully it's going to be quicker than last time, but let's see. So one example, the final outcome is going to be the connection of the spike glycoprotein with the receptor binding domain opened up, connected to the human ACE2 receptor that also has the same exact sequence and the exact portion, which is the receptor binding domain, right? So this model has this red one, this model has this red one. Could we use alignment to map them to each other without trying to manually navigate how do I actually fit them together, right? So that's objective number one. Objective number two is, okay, well, could we then take the SARS-1 ACE2 receptor and the SARS-2 spike glycoprotein, sorry, not the receptor, and align them to each other? Again, we have the models, PDB models for both. Could we use Chimera, what we'll use today, to map them together? And it's very simple, so hopefully all of you can follow along with me. Okay. So I have in our Dropbox, I have both of these session files for Chimera. These are the CXC. So I'm just going to open this SARS-1, SARS-2. Okay, and they look like this. So if you follow along, I'm sorry if you know, it is going to be different for maybe some of you. Some of you are not using Chimera. Some of you are maybe using Pymol. Some of you might, you know, have trouble opening this up. So I'm sorry about that. But hopefully this is something that later on you can watch the video again and try to do on your own. So again, I shared with you the Dropbox link to the file, which is this session. And if you open it, it should look like this. So please let me know if you've gotten there. So I know that at least some of you are able to follow along. Essentially what we're doing is going to reproduce what we tried doing last time. I had some difficulties last time doing it on my computer. It kept getting stuck. Okay, so please let me know if you're able to open the session. The session is called SARS-1, SARS-2 together. Please write in chat if you're able to do that. How can we map our sequence query with reference spike glycoprotein? Okay, yeah, so that's exactly what we're gonna see, right? So here we have, these are PDB files, but they also contain the sequence. So you can open the sequence viewer here. So this is the sequence. And these are two models. So you can see that the blue one and the red one, right? So these two are the SARS-1 and the SARS-2. We know that these sequences are fairly similar, right? We've already done the alignment part, just looking at sequences at nucleotide level and amino acid level. But now we want to understand, okay, we know how they map. How do they map structurally? So we just want to take a look at whether we can align them by looking at the sequence, and then also see how they align on this 3D part of it. And so we'll use it for this portion, and then, oh, okay. Sorry, it might be big, but I'll tell you how you can there are these individual files are there. Let's see if those are, okay, yeah, so this should be much faster. So in fact, I can show you how to do this. Okay, so if you open one of these CIF, which is essentially a PDB file, these are much smaller. Okay, so so let's just color this one red, right? You just type in here color red, and then you can just open the second one, 6ACC, just drop it on top like that. Okay, so there's your second one, right? Just to make sure that they're not, you know, on top of each other. I mean, you can see that they are kind of similar, right? So they open up very close to each other. Okay, So just try opening the individual CIF files. These are the PDB files that you can, they could be PDB, CIF, it doesn't matter. So just open them in Chimera. So we can take this blue one and just move it over right here. So we'll have, exactly the same thing. Okay, so we have these two files or these two sequences. Again, you should see something like that. And yes, it will give you a score. So I'll just show you in a second, very simple command, and you'll kind of see how they align. Okay, Athanasios, were you able to just open these individual files? So these are, okay, great. All right, great. So now everybody has it, right? So all we need to do is type in here match. And you can see that this is model number 1 and this is model number 2. So what you do is model number 1 to number 2. And you press enter. Okay, and you can see here, it's gonna take a little bit of time to do that, but here you have the result, right? So the command is just very simple, match number 1 to number 2. I'm just going to put that in chat. Okay, and what do we have? We have the two sequences aligned to each other, and we can now take a look at how they compare, what areas overlap completely and what areas do not overlap very well. Okay, so if you got the same result, you should be able to see here in the log window how, so how it was done. And we talked last time about Blossom 62, missing lose. Loops, do you mean the missing structure part of it? Yeah, so they both will have some missing structures. We looked at it last time where we saw how if you open the sequence, you actually have the sequence viewer, and some of the sequence that is known for each protein is not going to have, right? So right here, if you hover over this, you see no corresponding structure residue. So some of these do not have a corresponding atomic 3D structure. So that's normal. Okay, so this is, again, something that I want to get through quickly. So if you were able to get this, right, you'll have much more time to kind of study this. I want to also show you how the same principle can apply to the second problem, right? Because then I have a little assignment for you to work on if you want to practice and kind of investigate the outputs. First of all, before we continue to our next model, this specific model is actually you could do this iteratively. You can read more about this command and what it does. And what are some of the different options that you have for different situations. So this is a little bit more information on how this matchmaker command could be used. I want to quickly show you another option where you might want to. So here, what we did was this example, right? You can use this example with different types of multiple sequences or just one sequence. There's also more superimposition modeling component where maybe less similar structures could be used. But this specific problem also is very useful for the second example that I wanted us to try to understand how it could be used right here. The SARS, the ACE2 spike glycoprotein. Okay, so this problem, let me, so if you are, yeah, this is also unfortunately a big file. This is something that I have in the assignments as well, but I'll explain to you how I did this, and then hopefully, you know, it would be great to see if you're able to, you know, try and reproduce some of this analysis and look at the specific residues yourself, because it is time consuming, and I'm not sure that everybody is going to be able to do this. So again, here we have two separate models. You can see 6M17 and 6ACD, right? So I'm just going to type them. You can obviously, you now know where to find them, right? So 6ACD is the spike glycoprotein, right? So let me just show you here. If you search for 6ACD, this is the trypsin cleaved and low pH treated SARS-CoV spike glycoprotein and ACE2 complex, ACE2 free conformation with one receptor binding domain in up conformation, right? So this is one protein that you download, and you can see how, again, take a look at what exactly this sequence represents. This is in 2018, right? So this is obviously not SARS-2, this is SARS-1. And the second sequence is 6M17. So again, you can go to 6M17. And this is the 2019 novel coronavirus receptor binding domain ACE2 BOAT1 complex, or B0AT1 complex, right? So 6M17. So the objective here is, if you are just to download these two and load them into Chimera, what you'll see, and this is where it can get a little bit heavy, so I'm gonna hope I'm not gonna get stuck here. The problem is, you know, how do you know whether this spike glycoprotein really attaches in a specific way to the place on the ACE2 receptor, right? So what we'll do here, and I'm sorry that, you know, this might be a little bit fast and maybe not everybody can follow, but I'll just move this away. So in the spike protein, there are 3 chains with the receptor binding domain, only one will bind. Well, so what are we using it here in this example? We have, this is the ACE2 protein, but this model, right? So this is the ACE2 protein right here, but the model has also the B0AT1, which are these 2 orange parts. And also it has this receptor binding domain portion. So the receptor binding domain is already from the spike glycoprotein. What we don't know, or what I don't know, is how this portion that is also present in this protein actually causes the whole spike to attach to the ACE2 receptor. So in other words, this white part, and let's just change it. Let's just turn it on. Okay, so we have this blue part and the red part are essentially very close. They're a little bit different because one is from the original SARS and this one is from SARS-2, but they're very similar, right? Because both of these are called the receptor binding domain. Does that make sense, Dana? Okay, I hope it does. So again, what we do, we have here our model 3 and model 2. So what do we want to match? We want to match number 3, right, which is this, number 3 to number 2. Press enter. It's going to take a little bit of time. Okay, and there we go. Let's take a look. It's pretty good, right? So the overlap is pretty good. We have some results here. Again, we're not going to go into the details. Well, yeah, exactly. So the reason why we took this specific, well, not this one, but the previous one that we were looking at, this one, you can see that what they did is they put it into low pH. And what happens is that one of these RBDs opens up. And so you can see some of those, previous papers that we looked at where it talked about the ACE2 connecting to the spike glycoprotein is that the function, specifically in this novel coronavirus, is that only one of these will come up to bind to the ACE2 receptor, not all three of them. And so now we want to see visually, how does that actually happen? And so how does it happen? We load these two models together, we align them, and what we can now see is how they align between each other. Okay, so this would be the actual connection. So let's put it back into context. Again, I know that this is something that will require some more practice and some more time to think about both the outcomes and kind of how to prepare the models, and then looking at specific residues. So I have some assignments that I have for you if you would like to practice this more. Well, you align the whole model, but because you only have this exact same piece that is present in both models, only they will overlap. Right? If you were to use another command from this link that I gave you, look at that link again for Matchmaker. It has other options where it can also try to superimpose all of the models, and that's not what we want. In this case, we just want to find the matching ones and map them together. Okay, so this is what we wanted to do last time. We saw how these two models map to each other, right? Or align between themselves. So let's think about this ACE2 receptor. So the ACE2 receptor, this is the model. We have the hydrophobic and hydrophilic amino acids in this model. One clear example of how we know that this has an impact, this B0AT1 portion of this model is actually a part of the membrane. So we know that this is a critical part for it to be a part of that membrane. And now what we can do is we can actually apply some of these methods to study better how the different substitutions at the amino acid level in this receptor binding domain could have an impact on the affinity between the ACE2 motif binding motif to the receptor binding domain receptor binding motif, so RBM. Okay, so essentially the sequence level, at the sequence level right here, and you are correct that this is just one of the chains, but they are identical between all the chains, right? So here, once it opens up, so this red part is closed down. When it's activated, it opens up, and it kind of goes up and down as the conditions around it change, and it gets closer to the surface of the membrane. And then once it gets to the receptor binding or to the ACE2 receptor, opens up and binds. And so one of the questions is, how does that change the binding and how the different mutations in that portion and a very specific sequence that is right here, right? How do we know what changed about its affinity? And so there's a paper that we already discussed previously that can give us more insight into how specific amino acids in this portion that connects between the two, which is right here, the receptor binding motif from 424 to 490, changes in that specific position can impact the binding. Okay, and so this is something that we wanted to do last time, and I just wanted to quickly go through this exercise to make sure that we understand how the relationship between the sequence and the structure can help us understand some of these properties of the novel coronavirus. Okay, so now let's talk about some of the questions, and I kind of put up here some of the questions that can help us think about these things to make sure that everybody's on the same page. So did you have a chance to clearly see the significance of 3D structures for the study of infectious diseases? Did you understand how to download and visualize these 3D protein structures and how to work with them? Do you have now a better sense of how the 3D structure correlates with the sequence and the changes in the sequence, right? And whether you had the experience a little bit to alliances and 3D structures together. So if you can think about some of these questions, and if you have any questions related to these practical tasks that we just quickly did now and in the past couple of sessions, I'd be happy to address some of those. Okay, did anyone else have any questions about these tasks that we just did? Okay, so if not, I do have this assignment that will maybe help us understand whether you really, and I think you really understand um, once you practice. Yes, I will show this just once again. So let me just put it up here. What is the assignment? Hopefully by going through these steps, you'll be able to really validate that you know how to find the sequences, you know how to work with them in 3D, you know the relationship between the sequence and the structure, and then how you can align them both structurally and at the sequence level. Okay, so number one, find the RBM PDB sequence structure for SARS-1, SARS-2, and MERS. Yes, I would be happy to. Okay, so let me go back then to the questions and we'll answer some of these questions. Okay, so how can we find mutation point from the 3D protein or how we can make it? Okay, so remember, we have the sequence right here, right? So this is the sequence for chain C, right? So essentially any position here, right? Let's select some position. It's right there, right? So if we have the receptor binding domain, okay, you can see that it's right there. So it is actually between three. So let's go back right here. 306 to 527. I think it's a little bit different here. So that's an important part of it. So I think it's something like 315 maybe. Yeah, so 2, 527, which is 416. Okay, so it's right there. So this is the sequence of the receptor binding domain. Now, this is just the novel coronavirus receptor binding domain, right? So if, for example, once you do your analysis, right? So we had some analysis done. Remember that when you do multiple sequence alignment, the position numbers change. So you don't look for the number to be exactly the same. You look for the sequence to be exactly the same, right? Because there could be insertions to make the sequence align better. So you find some position. So let's say you found this sequence to be different. Okay, where is that mutation? It's right there. How can I see the impact of that sequence on whatever I am trying to study? Okay, you see that sequence is right there. Okay, maybe that sequence is, or that change is not going to be important, even though it's in the receptor binding domain. Maybe it's not important for the binding, right? So this is for SARS-CoV-2, yes, right? Because how do we know? When we go back here, right? This, well, sorry, this is SARS-CoV-1, not 2, right? But again, it all depends. So 6VXX is going to be the closed state, and I think I'm actually doing part of the assignment here. Okay, so you can see structure of the closed state, open state. So 6VYB, you could take 6VYB and 6ACD and compare those together, right? And then you will see how they align. Or you can take 6ACD and map it to this, to the ACE2, and then take the same thing with this 6VYB and 6ACD, right? And that's some of the ways that you could compare. But it gives you a sense of how you can actually work with both the sequence side of it. Again, if you identify a mutation, a difference, you can highlight and select it. And for example, you know, if I'm interested in this, I can do color cell orange, so it's clearly visible. Let's see. I'm going to okay. Oh, it's right there. Sorry. So there you go, right? You can identify a specific position and you know, make sure that you're looking at the right thing. Okay, great. So let's go through these questions one by one. Okay, so I can answer all of them. Okay, so let's go back to Athanasios' question, how to align. So again, the process that we're talking about here is we first do multiple sequence alignment, we see some kind of a relationship at the sequence level. And we see, for example, a specific domain that is of interest to us, right? So in our case, we found that by aligning, first of all, the whole sequence, the whole genome, we found that the most variability happens in the spike glycoprotein. We want to narrow it down. Where did we see that it has the most variability? It has in the S2 subunit of the spike glycoprotein. Which portions of the S2 subunit of the spike glycoprotein are the most important for binding? We know that it is the receptor binding domain. The receptor binding domain is this whole little piece that comes up. So if the whole piece is important, now we want to see which parts of that are important for the binding, for the affinity. Those are the receptor binding motif, which is just the interface between this portion right here and this portion right here. So how can we study that interface from the standpoint of physicochemical properties that are responsible for the chemical connections between these two molecules? We can look at a model that has the two pieces bound together, right? And just to get a sense of how that works with the whole protein, we found the open model with the protein. And so we know that the receptor binding domain of both models are actually the same, right? And so then now we can align those two together. How do we align those two together? We take one model. So let's take this model right here. I'm just going to select the whole thing. It's going to take a little bit of time for it to get selected. Okay, let's do the whole thing orange, and we'll move it. Okay, so I load the two models. I don't know exactly how they align to each other. I want to understand how do they align to each other, right? So what I do is I type here match. So I want to take just the matching sequences, not all of these models, just the matching sequences. And I say that there's a matching sequence between model 1, which is, or model 2, sorry, that this is this one, and model 3. So I will take model 3 and align it to model 2, right? So I just type this in the command, match number 3 to number 2. And I press enter. And you can see that it calculates, okay? So now it's connected, right? So how does it do it? It's matchmaker, that's the algorithm that connects chain E of number 2 to chain C of number 3, and it has a sequence alignment score of 8 to 8.8 and RMSD between 162 pruned atom pairs is 1.094 angstroms, right? And that's the area between these two, right? So how close they are. And in total, there were 182 pairs of atoms. Okay, so generally speaking, Athanasios, does that answer your question, how to align them? Okay, great. Now let's go to the next question. How can we find mutation point from the 3D protein, or how can we make it? Okay, so I hope I answered that question, right? Dr. Jasm, can you let me know if we answered that question already? Okay, great. Let's go to the next one. The 6M17 has both RBD SARS complex with ACE2. Yes, so 6M17 is the ACE2, the B0AT1, and the RBD from the novel coronavirus. Okay, Claudia, does that answer your question? Okay, Shaheen Alam, asking for a reference paper. So we did talk about the previous, in some of our previous sessions, we talked about, maybe I didn't put it here, 1 second. Okay, there we go. Yes, so here, there's a number of papers here that I highly recommend going through related to this topic. Okay, and let me show you, for example, this paper right here, which is where this image was taken from. It actually walks you through some of the considerations around the receptor binding domain. And the specific motif that is in the middle there, the RBM, receptor binding motif, and what specific changes occur there when it connects, compared to other ACE spike glycoproteins. So Shaheen, does that answer your question? Okay. Jethro, is it the spike glycoprotein sequence of SARS-CoV-2? Okay. Babis is asking, so assuming we use the software and find the sequence difference between two proteins, how do we know what is the significance of the difference? Okay, great question. So how do we know? That's exactly the point that we want to do next. So let me describe the assignment here next. And for Thurinda, the drop-down menu doesn't contain any subparts. That is actually something that happens after you start playing around with the different sequences. So for some reason, it first loads and only has the missing structures. But as you start digging through the sequence and playing around with it, it starts coming up. I don't know why that happens in Chimera, but eventually you'll see all of them. Okay, so let's go to, okay, does anyone else have any more questions about this? Okay, so here, I just want to, please, if you have any other questions or concerns about this, you feel like you don't know what's going on or how to do certain things, I'd be happy to address those questions. So here you can see the different mutations, exactly what was being asked by Babis. So please, Babis, let me know if this kind of gives you some idea of what we are trying to do here. So here you can see the SARS-CoV-2 receptor binding domain ACE2 and SARS-CoV receptor, so the novel coronavirus and the SARS-1 coronavirus interfacing with the ACE2. And you can see that the contacting residues are shown as sticks at the SARS-CoV-2 RBD ACE2 and SARS-CoV RBD ACE2 interfaces. Positions in both RBD that are involved in ACE2 binding are indicated by red labels. And sequence alignment of this contacting residues are indicated by black dots. Contacting residues by SARS-CoV RBD are indicated by red dots, right? So the first one is red dots. The second one are the black dots. So if you just look at the sequence alignment, you want to see how different black dots are from red dots, right? So what is the difference? Structurally speaking, what is the significance of some of these mutations? Is it because they're just different? Or is it because as a result of these changes, different residues contact with the ACE2, right? Because the ACE2 doesn't change, it's still human ACE2. So it must be the viral protein changes that are responsible for how differently it binds. So here you can see the specific residues that they have identified, and I propose that you try to identify them as well by doing multiple sequence alignment. And so here you can see the black ones are coronavirus 2, The red ones are coronavirus 1. So some of them are exactly the same, right? Some of them are different. For example, this G and T, right? That's a change. As a result, the T does not bind anymore to the ACE2, right? Here, this one binds, but this one is an N, it does not bind, right? So think about it. Remember, as we talked about different physical chemical properties, the position, the 3D structure, all of these characteristics explain how that change actually occurs. Okay, so that's to answer Bobby's question. And let's look at the proposed assignment. Again, nothing that you have to do, but if you're getting how this works, I think it's going to be a very useful exercise for you to feel like not only you've seen somebody else do it, but you can do it yourself. So number one, find just the RBD PDB structure for SARS-1, SARS-2, and MERS. Number two, align the 3D structures and find differences between them. You can do this using multiple sequence alignment. You can do it in Chimera or PyMol by aligning the 3D structures. Explore what these differences might mean for ACE2 receptor binding. And obviously, that also means look at the paper and you can see some examples, or all of the papers that I shared with you, you'll see some examples of how people use this kind of alignment and superimposition to infer how those, how the affinity changes and what might be the impact of that. Okay, so any question about this assignment? Okay. So I do have, yes, and all the videos we will post on the forum, Dr. Jason. Bibsha, if you could please check if session 7 video is up. And maybe post a link here to the forum as well. Okay, so I did have something that I wanted to talk more in detail about today. We don't really have time. I don't want to start a new topic because we spent a lot of time talking about these different issues. Okay, so there's a question: when we align 3D structures of protein by MSA, then output is phylogeny. Well, when we align structures, the structures, you can't really align structures. You can superimpose structures, and typically you would superimpose one on the second one, right? So just two. When we do alignment, we do alignment of sequences, and these could be nucleotide sequences or amino acid sequences. We can incorporate the significance of the change between those sequence elements, like nucleotides or amino acids, to try to understand how they are phylogenetically related or how they are evolutionarily related. So this is something that you can use to build a phylogenetic tree. Does that make sense, Manasvi? Okay. Okay, so I'll briefly tell you what I will cover hopefully next time. We wanted to talk about some other aspects of mutations, not related to structure, right? So having a sense of how mutations impact structure is important, especially for structural proteins. As we know, not all proteins are structural, and mutations occur across the whole genome. And to help us think about this, to help us think about this, we will use in our next session polio as one of the viruses that has been studied extensively to understand in the context of vaccine design, as well as the disease progression, to understand how mutations across the whole genome might have an impact on virulence and have an impact on what's called mutational fitness, or you can call it fitness of the whole virus, evolutionary fitness. So what does that mean? I just want you to kind of be able to play around with this tool and make sure that you understand kind of what's going on here. We want to talk about different types of mutations. As you can see that these types of mutations are all listed up here. What properties do we know of these mutations? There's rate of mutation, and rate of mutation can help us understand how often these mutations happen. For example, positions that have multiple changes in them across a number of passages. These are passages. That means that those are rapidly changing, right? Those that are conserved, their rate of mutation is low. The ones that are conserved, right, so all the gray ones, they might have specific implications for the virus. If the virus does not change them that much, that means maybe they are more important. We also have synonymous and non-synonymous, so those that change an amino acid or don't change an amino acid. And we can play around with this here. And we have the fitness component as well. And the fitness helps us understand whether these are beneficial for quasi-species replication and composition. What are quasi-species? Quasi-species are groups of very similar genomes that all occur in a passage, right? So a passage is a group of cells where the virus is grown, it replicates, and then whatever ends up getting out of those cells is taken to the next passage, etc. We'll talk a lot more about this next time, but I just wanted you to take a look at the tool and play around with it to think about these different types of mutations. So what are beneficial mutations? Beneficial mutations are the ones that accumulate over time, over passage time. This is a tool called Sequence Navigator, SeqNav. It's essentially an output from the Bioinfo platform. You get a visualization using this tool to characterize these viral mutations, but for very specific type of sequencing. So here you can see beneficial mutations, right? Beneficial mutations are essentially the ones that are needed for viral fitness. If you think of an athlete and you change shoes every day to check which shoe is the best, and you find a shoe that works for you best all the time, you will pass it on to your son or your daughter to run with those, and that would be a good representative of a beneficial mutation, a shoe that works really well. If they are detrimental, that means that that doesn't work. These mutations disappear over passage time. If these are lethal, that means that that whole genome that accumulates mutations in these positions will not survive to the next progeny. And if they are neutral, that just basically means that this is some kind of a stochastic process. These appear, disappear. There's no really connection between the viral survival or ability to replicate and produce progeny with those mutations occurring in its genome. So now we can map these again on the structure. So for example, here we have different polio proteins, and we can look how these different positions could be mapped on the 3D structure. So let's turn all of these on. Okay, so this is what we see here, and we're going to color by fitness. Okay, and here you can see the different types of mutations. You can't see this page. This is the link. Please try using Chrome. Okay, can mutations be induced strategically that will be lethal for the virus? Yes, and we'll talk about vaccine design a big part of vaccine design is using what we understand about specific mutations to attenuate the virus, right? To make sure that the virus is contained within a certain tissue or replicates only at a certain rate, or even could be, essentially just replicating for a very short time until it completely is dead, right? Just enough time to train the immune system. So we'll talk about that in the context of vaccine design. Okay, so Athanasios, I'm sorry to hear that it doesn't work for you. Does anyone else experience this problem? Maybe you can share a screenshot for me to look at for the next time. Okay, so please take a look at this. Again, we don't have to look at all this now, because I understand that there's a lot of material that we already covered today, and I want everyone to have a chance to play around with the 3D structures and understand the structural impact of these different mutations before we jump into, essentially, these are different types of ways to characterize mutation variants, but they're all important for our understanding of the viral evolution, of replication, of how it is, good at what it does, causing disease. And studying those aspects with a model virus like polio is great, because there's a lot of good data. So if you are interested in finding this data and reading a little bit more about this, obviously you can just copy this over to NCBI and find those, right? So if you go to NCBI and you look for this SRR, SRR 1036, okay. Not sure what happened, but I will share with you the paper that all of these data sets could be found in. And yes, Claudia, we will actually take a look at the biological process of how these mutations are discovered. These are low frequency mutations, so they require specific library preparation before the sequencing is done. These are then checked in phenotype, what kinds of replication rates and virus titer they produce if they accumulate over time. So this is biologically validated, and I'll share with you the paper for next time as well. I don't want to move away too far from what we did today with the structural analysis, but we will talk about this extensively in the first part of our next session. So briefly speaking, in conclusion of this session, because I do have something coming up, I wanted to come back to this problem of your own independent project, because we're getting very close to that part of the program. So as you remember, an independent project can start with analyzing somebody else's data, where we take published and digested data and methods, reproduce the same result, and you will see that you will start having a lot of questions about the same method applied to different data, the same data but different methods, and how you can essentially then transform a practice analysis that you've done with somebody else's data and methods to something of your own. And that's really the goal of this program. So last time we talked about how we can take this idea and apply this to design your own project, right? So here I gave you example last time of how I could think of, different questions appearing in my mind, dealing with these different sequences, and how I could start collecting publicly available raw data for me to do the analysis, reference publications that explain the significance of the problem that I'm trying to solve, and also translate my question, my hypothetical question or conceptual question, to some kind of a practical analysis or study. Okay, yeah, so if you have any questions about filling out that form, and again, I'll just paste the form here for everybody to take a look at. So this link, if you go to this link, you will see a form. You should have your name and email address and things like that pre-filled out. But then you want to fill out here the problem statement, the project summary, the link to the raw data, which might be a bio project, and then some reference publications. Okay, Manasvi has a question. Is the project only about infectious diseases by virus only? No, you can also do any kind of infectious disease. You can see, for example, you know, bacteria, right? Right. Okay, so here's another suggestion, Ryan. For project ideas, are there any current COVID-19 phylogenetic risk analysis studies? A patient with T2D, CHF, and infected with COVID-19, a patient who has a heavy smoker who is diagnosed with COPD with COVID-19. Yes, so those are excellent questions. As you can see in the example that I was showing, you know, an interesting, maybe to me, right, question was how immunocompromised patients, so patients that have immunosuppression drugs, right, those are that have autoimmune diseases, for example, respond to different viral infections, including COVID-19. So I have some reference papers here, and they all have some patient data available. So if you're interested in any of those, you know, one of the main challenges here is, can you find sufficient data to do such an analysis? I mean, obviously you can do multiple sequence analysis. Question is there data, right? Okay, I'm most interested in Plasmodium falciparum causing severe malaria in humans. Well, great. So if you can take you know, again, and find data, right? So you take this, you go to NCBI, and you have to not only find data, right? You also want to find specific type of data that you want to focus your question, right? So if you're interested in, you know, malaria in humans, can you define a very specific question? What do you want to study? As you can see, there's, you know, gene expression data. There's all kinds of information. There's nucleotide data. There's all kinds of information that you can find. What specific question are you trying to answer? Okay, so this is a challenge. It does take some time. It does take some exploration of data sets and reading through literature to identify a good question. So that's why I'm trying to help you start thinking about this starting from now. And if you want to see how similar projects were developed by others, we do have a section on the platform that's called projects. They're not all going to be related to viruses. There's different types of projects, but you can search through the projects to see what else is there. And we are adding new ones. In fact, during this program, we've been adding projects on COVID-19 and Ebola and EVD68. So by the time that your time to work on projects starts, you'll have those projects as well. And we've used some examples from those projects in the sessions that we did. And all of these should be free to the participants of the program, but some of them you'll see coming soon, and they will be available starting from when the project time for this program begins in a few sessions. Okay, so thank you again for joining. Our next session is going to be on mutation, annotation, and significance. So we'll kind of complete this portion with polio. Okay, the deadline for project idea submission, it's up to you. I mean, we talked about different levels of certificates you can get for the program. If you complete a project and you want to have enough time to work on the project, we suggest that you submit your project idea for review after June 4th. Yes, obviously the data has to be public domain because you will be analyzing it. You know, all of your pipelines are going to be publicly visible. We will all see your analysis, right? So you want to have it publicly available. Okay, great. Well, thank you for joining today. I'm looking forward to seeing you again soon. And please, if you have any additional questions, post them on the forum. Thank you very much.